Thank you so much for letting me present our research work on social feeding behavior as a novel measure of assessing social motivation in dairy heifers. And my deepest apologies for not being able to present this work in person. Before I begin, I would like to respectfully acknowledge that the work presented here was conducted at the UBC Dairy Education and Research Centre in Canada, which is situated on the traditional, ancestral, and unceded territories of the Hekolekim-speaking Coast Salish people, including Stalo and Squaxin Seaboard Island First Nations. Why do we need to be concerned about social motivation in dairy cattle? As gregarious animals, cattle are highly motivated to maintain social contact with conspecifics. However, individuals may differ in their preference for social proximity. And that means in confined housing conditions, which most cattle are kept in, individuals might not be able to live according to their social needs. And this can result in social stress, for example, competition and negative social interactions during feeding. So in order to provide all animals with a good life, it is required that we understand the needs of the individual. Social motivation is a concept that is well studied in cattle. Traditional tests involve separating the test animal from its group and measuring its behavior. For instance, in the runway test, the time an animal takes to reunite with its penmate is thought to be a reflection of its social motivation. But given that isolation and constraint are aversive situations for cattle, their response is likely to be intertwined with other internal factors of the individual, for example, fearful personality traits. Of course, social motivation has also been studied in the presence of conspecifics. Proximity-based social preference or inter-individual relationships based on affiliative or agonistic interactions are common approaches to derive individual variation in social motivation within the group context. However, the reliable assessment of these social behaviors can be very difficult. And more importantly, when assessing the preferences of animals in confined conditions, we need to keep in mind that animal behavior is most likely to be constrained by that confinement. Hence, the observed behavior might not be aligned with the preference of these individuals. Therefore, our study aimed to develop novel social motivation measures derived from testing situations providing animals with greater choice over social proximity in the feeding context. We used feeding situation as testing environment because of the aforementioned negative impact of stress when social needs are not met. And since we developed novel measures, we also wanted to compare the relationship to traditionally taken measures in social isolation. Our experimental design was the following. We tested 36 heifers, and we first exposed each heifer to a series of standardized isolation tests. We conducted a runway test, which I've already explained, and we also exposed each individual to 10 minutes of being socially isolated in an unfamiliar arena and measured its behavior in the novel arena test. We conducted the social isolation test 14 days later, expecting that in line with personality traits, the behaviors that we here record would be consistent across time and context. Each individual was then also tested within a group context where we designed two novel social preference tests, a social feeding test and a social feed trade-off test, which I'll explain in more detail in a minute. And our hypothesis was that if all of these different tests measure social motivation, then we would expect a significant relationship between the outcome variables of these tests. We designed a novel social feeding test in which we tested heifers in groups of 3, and we tested 12 groups in total. Animals were put into a testing arena that consisted of 2 troughs that were placed along one side of the pen, and the 2 troughs were separated by a physical barrier that prevented visual contact to the other side. All animals were habituated to move between troughs and feed from both sides prior to testing. and animals were not food deprived for the purpose of testing. During testing, we applied a very specific feeding schedule, where one side of the troughs received twice as much grain as the other side. We applied this very specific feeding schedule under the assumptions that animals would distribute themselves according to research availability, also known as idea-free distribution theory. So by providing twice as much feed on one side than the other, and animals knowing that feed is available on both sides, we anticipated that the group would split up and one animal would move over to the other side to feed alone. Hence, we expected that the time an animal spent feeding alone could be a reflection of social motivation. In fact, social motivation has been assessed this way in free-ranging animals, such as sheep, but the concept of idea-free distribution has not been applied to intensively housed cattle. So we measured the distribution of the heifers within 10-second scans intervals during the 5 20-minute test sessions. To test social feed trade-off willingness further, we tested the same 36 heifers, but this time individually. Heifers were placed into a testing arena that consisted of feed buckets containing grain that were placed at a cruisingly distance to conspecifics, held in an adjacent pen with visual contact to the test animal where this animal was feeding from the first bucket. And when we measured the latency for the animal to leave this area and move on to other buckets that were placed at a cruisingly distance, and they were reducing the visual contact to conspecifics. We measured the latency for test animals to leave each of these bucket locations and move on to the next bucket, expecting that the latency to leave and move on would be a reflection of animals' willingness to move away from companion in exchange for feed, as has been proposed in previous studies. All heifers were tested twice. In the social conditions, test heifers had visual contact to conspecifics while feeding from bucket 1, and we also tested each heifer in an alone condition where the companions were moved behind a physical barrier, preventing visual contact to the test animal. Preliminary analysis showed that the conspecific presence had a significant effect, but only when leaving bucket 1, with latency to leave being significantly longer in the social condition than in the alone condition, indicating that this measure is a sensitive measure for social motivation. And then we used the difference in latencies to leave bucket 1 in the alone and social condition as a trade-off score for further analysis. Coming to the results, we first compared the measures taken in social isolation and social feeding test. First of all, we found, looking at the measures taken within the social isolation test that were conducted 14 days apart, that behavior showed very low repeatability, except for vocalization and runway latencies. People familiar with per-se tests might have expected us to do a principal component analysis. However, our data was not suitable for this type of statistical analysis because of the low repeatability of the data. So moving forward, we only used vocalization and latency to reach peers as measures of social motivation, and compared this with the social feeding measures, where time feeding alone varied significantly among individuals, with some animals never feeding alone and others feeding for more than half of the test time alone. However, there was no association between the measures we've taken in social isolation and the time spent feeding alone. It should be noted that groups did follow the expected 2-to-1 distribution for over 80% of the test time, indicating that the test setup that we used for the social feeding test did encourage idea-free distribution. However, we also had a group that never split up, and despite animals moving between troughs, they always moved together and never split up, possibly suggesting strong social motivation. We also found no association between the social isolation measures and the social feed trade-off score. And finally, we compared social motivation measures in the same social context. So here we did see large inter-individual variation in feed trade-off willingness, meaning that animals with negative values had greater reluctance to leave their companions in exchange for food. However, comparing this with the time spent feeding alone, we found no relationship between measures. From this, we can conclude that measures taken in the social feeding test did not correspond with animals' responses during social separation. And this is in line with previous studies showing that social feed trade-off willingness Thank you all for your attention, and my co-authors will now be happy to answer any questions.